So, hello students. So, today let us do this topic on muscle contraction. So, as we can see in the name itself, the important property of muscle is written. That is, it is muscle is contractile in nature. If a nerve, for example, is excitable in nature, muscle is contractile in nature. That means it has the ability to shorten and then come back to normal length afterwards. Under certain circumstances. So that is the property of contractility, which is specific to muscles. It's a unique kind of cell. Now, you may have learnt in the earlier classes that we have got one type of muscle called as smooth muscle, another type of muscle called as cardiac muscle, and another type of muscle called as voluntary muscle. This voluntary muscle is also called as striated muscle, and therefore the smooth muscle is also called as non-striated. So this particular discussion is with respect to voluntary muscle or striated muscle. So voluntary or striated muscles are attached to bones, so they are also called as skeletal muscle. So always wherever there are two bones and the bone needs to move, and there we have voluntary muscles or striated muscles. Whereas smooth muscles, for example, they are there in the bladder or they are there present in the alimentary canal. So these are non-skeletal muscles. Cardiac muscle is present in the heart. So this is, now if we this is a typical muscle, how it looks like, and this will taper towards one end, and there will be, this is where it is going to attach to the bone, tendon over here and tendon over here on this side. If you take a small part of this, so we will find that the muscle is made up of several bands, what we call as fasciculi. So these are actually made up of muscle fibers or cells. If you take a look at a part of this, and we are seeing a muscle cell, under a light microscope, and we will find these so we are, as I told you, restricting ourselves right now, talking about voluntary muscle or striated muscle or skeletal muscle. So these muscle cells are long, hence they are called as fibers. Just like we call nerve cell as a nerve fiber, similarly muscle cell is often called fiber because it is very long. These are non-tapering. Because when you're seeing it under the microscope, a light microscope, they are quite long, therefore you don't see the ends tapering. So that's why they are non-tapering. And we see striations. So these are the striations. These striations are called as alternate because they look dark and then there is a light. Dark and light bands are there. And another thing that we often see is that they appear to have several nuclei. There are several nuclei, a multinucleate condition. But these nuclei, so suppose we take a cross section over here. The nucleus is not in the center but in the periphery. The nucleus is in the periphery. And we always use a term, a prefix called as sarco for muscle. So the plasma membrane is called as sarcolemma. Its endoplasmic reticulum is called as sarcoplasmic reticulum. Its cytoplasm is called as sarcoplasm. So the outer plasma membrane is called sarcolemma. The sarcoplasmic reticulum means endoplasmic reticulum and sarcoplasm means cytoplasm. So everything is, there is a prefix called sarco. That's why cancer of the muscle is called sarcoma. So these are some terms. So now let us look at these alternate light and dark bands. Right, so what we have drawn is the part of this myofibril. So these muscle cells are made up of myofibrils, those striations, and we said that there are two bands, dark and light bands. So dark band is made up of myosin, light band is made up of actin. These are proteins. So this dark band is dark because light doesn't pass through it easily. It gets refracted. So this is called as anisotropic. Whereas the light band, the actin, is such that the light passes through it easily. So it is called as I for isotropic. So I for isotropic and A for anisotropic, because light passes here, light doesn't pass over it. So these are also called as I band and A band. So the dark band is called A band, the light band is called as I band. So here we see that all these purple colored structures, they make up the protein myosin, and therefore this is called as A band. Whereas this entire part this is called as I band. So they overlap each other. I band and A band overlap each other. So I and A bands overlap. Just like we put fingers of two hands into each other and you get an overlap. So similarly you get an overlap. If you push them too far, the overlap will be too much. If you push them less, the overlap will be less. So something like that is taking place over here. So we have an A band here and we have an I band. Okay. Now the center of the I band over here, this is like a disc-like structure. This one is called as Z line. Similarly, there is another Z line over here. The distance between one Z line to another Z line is called as sarcomere. Sarcomere is unit of muscle. So we have terms like sarcolemma, sarcoplasmic reticulum, sarcoplasm and sarcomere, unit of muscle. What is this unit of muscle? From one I line to another, from one Z line to another Z line. So it is containing one part of the I band, a full A band, and one part or one half of the I band. So partly it is light band, fully dark band, and again partly I band. That is called as one sarcomere. So from one Z line to another Z line. This is how it is looking like. Now when we say that the muscle contracts, it means here I will briefly show you. I will not draw the entire diagram, but briefly this will look like so the Z line, one Z line will come over here and another Z line has come over here. That means the entire muscle has pushed this way. See, therefore from here this has gone inside. The length of the A band is remaining constant. Length of the A band. So here contracted muscle and this is a normal relaxed muscle. So see, what has happened is that it seems that the I band or the light bands have slid into the A band. Therefore, the length of this remains constant. So during contraction, A band length is constant. I band slides into A or into myosin. This is why this is called as sliding contraction theory. Now to understand the molecular mechanism of this, here itself we can show so first let us write about myosin. This is myosin head, and this is the myosin tail. So there is a two protein chains are there, and at one end it will have a myosin head at 90 degrees to this long axis, and here this has got a site where ATP can fix inside, ATP site. So myosin is made of two chains, and head has got ATP site, and it has got the enzyme where it can break down ATP. So this function is part of myosin. Whereas actin is made up of, when we talk of them as individuals, they will be spherical. So they are called as G protein, G for globular, but when they all assemble together, it becomes F actin. They become F actin. So when they are separately in a circle, it is called as G actin, but when they all assemble together with these spherical structures, it is called as G actin. Now actin molecule also is arranged in such a way it has got a thread-like structure that is moving everywhere. This thread-like structure is another protein called tropomyosin, and this tropomyosin this is troponin. Those spherical molecules are troponin. So F-actin, you can think of it as a string of such marble-like structures of G-actin that make it fibrillar. Within that there is a thread-like structure called tropomyosin. Tropomyosin also has got individual molecules called troponin, and these troponin molecules are sensitive to calcium ions. They can bind to the calcium ions. Now normally, so let us now look at how this is arranged together. So these two will come much closer to each other. I am showing over here a myosin molecule with an ATP head. There will be many such myosin molecules. See, some will be attached like this. Look at the events or the physiology of this working. Under normal conditions, that means under normal conditions of rest, when it is relaxed, myosin head is bound to actin. Myosin head is bound to actin in the way that I have just shown over here. Myosin head is at 90 degrees to actin, as I have shown over here or in the earlier place. This angle is 90 degrees. This particular angle is 90 degrees. So it is bound to. However, when we say that they are bound, they are not actually completely bound. The reason is that tropomyosin prevents proper binding. But when muscle is stimulated by nerve, when muscle is stimulated by nerve, at the neuromuscular synapse, calcium ions rush into muscle cell. Calcium ions bind to troponin. Troponin pulls tropomyosin. Tropomyosin fits into groove and therefore allows myosin head to bind to actin. So look at this particular part over here. Here the tropomyosin is in such a position that is not going to allow this to bind to the actin. But suppose this has fallen into a groove, into the inner groove, and it is allowing it to bind to it. And that is the difference between, here I have shown the binding, but actually they have not bound because of the tropomyosin. So let us repeat, these are the steps. Under normal conditions, actin and myosin are associated with each other. although we are saying that they are bound to actin, there is a certain distance between the two which doesn't allow them to bind. And remember that the myosin head is at 90 degrees to its own tail. And the reason why the proper binding is not being allowed is because tropomyosin is there, is a molecule which is there. Now suppose the muscle is stimulated by nerve, then at the synapse, then calcium ions will rush inside because of the depolarization. And because of that, now the calcium binds to troponin, because troponin molecule is associated and is very sensitive to presence of calcium ions. Now when this takes place, the troponin pulls this tropomyosin. Tropomyosin now falls into a groove. And when that happens, it allows the myosin molecule to bind to actin. So actin myosin complex is formed. Now the myosin head has got ATP. ATP breaks down to release ADP plus energy, and this energy is used to turn head at an angle of 45 degrees. So I am showing it over here. Look at the angle now. It has turned at an angle of 45 degrees. Now when it is turned at an angle of 45 degrees, remember where was this located? This was bound to the actin. So this green structure which you have shown over here, this was bound to the actin. So let us draw that part once again over here. It is the actin, same molecule which we are showing over here. So when it was attached over here, and if it becomes now 45, that is the movement of this part, we can show with the orange pen, and in a dotted fashion it has moved along this direction. That means the entire muscle is going to progress and move in this direction. So if you come back to the original diagram, when we say that shortening has taken place, it means that see, because this muscle head is going to pull the actin here, this muscle head is going to pull the actin over here. So they are going to pull each other because of this myosin head. So the whole structure, that the actin therefore pushes into the myosin. The actin pushes into the myosin. That's why look into this diagram. These are all actin molecules. The actin molecules will push into the myosin. Similarly over here, actin will push into the myosin. That is why the length of the sarcomere has now become less. That is what is called as contracted muscle. In the normal case, this was the length, but when it is now contracting, it means this is moved inside. So this is how a muscle cell will work. With that, we come to the end of this topic. Thank you.